Thank you, Adam. Hello, everyone, and a warm welcome to all of our attendees. Thank you for joining the Bioprocessing Next Leadership Summit and Expo. The topic for this session is monoclonal antibody early development and critical considerations for commercialization success. The agenda for the next hour will be as follows. First, I'd like to give a brief introduction on antibodies and monoclonal antibodies, just to get everyone, particularly any individuals who may be new to the bioprocessing world, baseline on what they are and what they do. Next, we'll talk about the therapeutic history of monoclonal antibodies, as well as get a glimpse into their contribution in the current global drug development pipeline. Third, we'll cover the market ecosystem, specifically the drivers and factors affecting their development, as well as discuss the overall challenges of bringing a target molecule to market. Fourth, we'll cover some high-level regulatory considerations to keep front of mind, especially during the first-in-human trials. Next will be the early phase from DNA to IND or IMPD filing, and explore some principles that can be employed to accelerate early development success, and how we as a contract development and manufacturing organization approach it, and share a case study. After that, we'll turn our attention to the considerations for the late phase and commercialization and share a case study as well. Lastly, we'll wrap up and have a brief Q&A portion. To get us started, I'd like to pose a few questions and cover those in this section. What is an antibody? What is its function? And how are they produced? Antibodies are protein molecules which traditionally have a characteristic Y-shaped appearance, and depending on their particular class, can be found circulating in the blood, in tissue fluids, in mucosal membranes, or secretions, and in concert with other cells and pathways of the host immune system, act as a sentinel to recognize and protect the host from foreign organisms. B cells in particular, a type of lymphocyte, is where antibodies are produced. The antibody-specific protein sequence, along with other post-translational modifications, creates a particular 3D structure which binds like lock and key to antigens, which are structures on the outside of foreign substances. Antibodies have what are known as hypervariable regions, which contribute to their immense diversity and selectivity and form a pocket to interact with antigens. Antigens can be proteins, lipids, carbohydrates, or nucleic acids, and include viruses, bacteria, cancer cells, and even common allergens. Once antibodies bind to a particular antigen, the immune system responds to neutralize the foreign entity. This antibody mechanism is leveraged against infectious disease, whether by natural infection or vaccine-mediated immunity. Polyclonal antibodies are not identical in sequence or structure and target different areas even against a particular antigen, whereas monoclonal antibodies, or mAbs for short, are identical copies derived from a single clone or parent B cell and share identical protein sequence and thus structure against a particular antigen, and recognize the same antigen with equal affinity and biological effect. However, in combination with recombinant DNA technologies, engineered antibodies can be used to treat cancers, inflammatory diseases, and autoimmune illnesses, and much more. This illustration on the right shows how engineered antibodies employ a variety of mechanisms and pathways to accomplish their objective. These engineered antibodies recruit other cells of the host's immune system to mount defensive measures, typically with less off-target effects often associated with traditional small molecule drugs. Antibodies can also block or otherwise disrupt cell surface receptors and corresponding signaling as a mechanism to modulate disease-related pathways. Alternatively, antibodies can target soluble molecules such as plasma proteins and either block or sequester them from regular operation. Some monoclonal antibodies called ADCs, or antibody-drug conjugates, are designed to carry a drug, a radioactive agent, or another molecule and deliver that payload to the cell targeted by the antibody interaction. There are also other monoclonal antibodies called bispecific antibodies, which are engineered to recognize two different antigens and thus bring different target cells into close proximity of each other for an intended biological effect. mAbs are a potent, effective, and proven therapeutic in modern medicine, but we'll focus specifically on traditional mAbs in today's presentation. Therapeutic monoclonal antibodies are the output of complex biological processes. Simplistically, the production of a mAb has both upstream and downstream procedures. The upstream process deals with creating the ideal cellular factory and includes cell line development, clone selection, media optimization, growth, and scaling up from smaller bench scale to larger bioreactor volumes, and finally, the harvest and collection of the cells. The goal of the upstream process is to leverage the cell's innate ability to produce the desired functional monoclonal antibody with maximum output in minimum time. The downstream process serves to isolate and purify the desired monoclonal antibody from the cell's environment with maximum purity and minimum loss. The entire process is supported by quality control steps, automation technologies, and analytical methods to ensure reproducible output and biological activity from run to run and as the scale increases. mAbs are a product of the process which relies on a large number of material inputs, instrumentation, procedural steps, and technical decisions. Thermo Fisher is one supplier offering an expansive portfolio along the continuum of bioprocessing, from media to mixtures, cell lines to centrifuges, resins to refrigerators, and much more. In this next section, the questions being addressed are, what part have monoclonal antibodies played in human therapies and for how long? What level of contribution do mAbs play in the current global drug development pipeline? And what does the ownership profile of these molecules look like? Since the FDA's initial approval of its first therapeutic monoclonal antibody back in 1986 to treat organ transplant rejection, the number of mAbs has exceeded 100 different approved molecules as of 2021, with multiple molecules undergoing regulatory review. While cancer remains the primary indication treated by monoclonal antibodies at nearly 50%, the breadth of indications has expanded to include neurological diseases, genetic diseases, and infectious diseases like SARS-CoV-2, just to name a few. A large portion of the top 20 therapeutics by annual total sales is accounted for by mAbs. Well-known therapeutics like Humira for rheumatoid arthritis, Keytruda and Opdivo for various cancers, and Stelara and Remicade for psoriasis and digestive disorders are just a few of the billion-dollar monoclonal antibody blockbusters that are both life-saving and life-changing for countless people worldwide. Global cumulative sales of mAb therapeutics are estimated to be north of $100 billion US in 2021, and projections into 2025 are expected to be greater than $150 billion US and growing double digits annually. A 2022 Thermo Fisher analysis of Pharma Intelligence's Pharma Project Day 2021 database, which represents the global drug development and research pipeline, indicates mAbs as the most common modality in the overarching biologic segment when compared to a host of other biologic molecule types. In the preclinical phase, shown in the bottom left blue section, nearly 1,000 monoclonal antibodies comprise almost 40% of the potential biologics in development. In the clinical trials phases shown to the right, mAbs account for 40 to 45% of the overall pipeline by number, with almost 300 monoclonal antibodies in phase 1, 266 in phase 2, and 162 molecules in phase 3 trials. While bispecific monoclonal antibodies and antibody drug conjugates showed the greatest increase year over year from 2020 to 2021, traditional monoclonal antibodies continue to contribute significantly in the global therapeutic pipeline. Another takeaway from the analysis is the ownership of the preclinical and clinical pipeline is significantly owned by small and emerging biopharma companies, as shown in the chart on the left. Small and emerging biopharmas own 90% of the preclinical biologic molecules, some 2,300 plus compounds, and approximately 70% of the biologics in the clinical trials phases, which again includes significant representation by monoclonal antibodies. These small and emerging companies lack the capital, advanced development capabilities, and the manufacturing expertise, and thus frequently partner with CDMOs like Thermo Fisher Scientific to develop and commercialize their molecules. On the right side chart, speed of development and speed to market has been increasing as the number of drugs approved with accelerated regulatory designation has increased. The desire to fill unmet needs for serious medical conditions has brought about various regulatory enhancements over time, including accelerated approvals, priority review procedures, improved communication with health agencies, as well as leveraging surrogate and/or intermediate clinical endpoints to bring more effective solutions to market more quickly while maintaining stringent safety standards, regulations, and improved clinical benefits. In the last decade, the number of drugs approved under under an accelerated designation has climbed from roughly 50% to the mid 70% mark. In light of the position mAbs occupy in the global drug development pipeline, what other outside drivers are affecting the ecosystem of mAb development? And for that matter, the overall biologic segment? What specific market forces are affecting sponsor companies? And lastly, what are some inherent phase-specific challenges in developing and commercializing a monoclonal antibody? Let's dig into those questions here. The mAb development ecosystem is complex and contains various opposing drivers. First, speed matters to innovators because a human patient is ultimately waiting for these life-saving drugs. Patients, especially those with rare conditions, need new and more effective medicines faster. The small and emerging biotech company is particularly under time pressure to go from DNA to IND or IMPD filing so they can show investors progress is being made, which in turn allows them to secure additional funding based on milestones achieved. Second, the biologics development pipeline is growing more complex. Specialty drugs, including biologics, cell and gene therapies, and other first-in-class new modalities are increasing, not decreasing, the intricacies of how biologics are developed and manufactured. CDMOs are frequently being tapped to provide this technical capability to reduce the learning curve and to accelerate the path to commercialization. Third, while small and emerging pharmas may be fighting year to year to survive, large pharmas are not immune from their own financial challenges. They have seen declining R&D return on investment over the past decade. Large pharmas are supplementing internal discovery through mergers, acquisitions, or partnership deals with small and emerging companies, or even other mid to large size pharma companies to drive the speed to market and maximize what earning potential can be had. Fourth, the COVID pandemic demonstrated the important role a trusted CDMO or CMO can play in helping deliver product urgently. Whether outsourcing a lower priority project so the innovator can focus on higher priority programs, or leveraging the contract partner's agility and capacity to deliver drug substance and/or drug product. The pandemic also highlighted the need to evaluate multiple sourcing streams to both meet demand and prevent unexpected supply chain deficiencies. Lastly, the increasingly digitized world we all live in is also transforming the pharma ecosystem. Big data is driving workflow insights, AI and machine learning are aiding molecule design and prediction, and increasingly interconnected digital data systems are improving efficiency, decision-making, real-time visibility, and innovation. In summary, there are multiple changing dynamics, applying pressure to all the players involved who are trying to enable new therapeutics in less time. To put a point on the aforementioned challenge of ROI on R&D spend, looking at the 10 years preceding the COVID-19 pandemic, a 2019 Deloitte report indicated R&D returns of 12 large-cap biopharma companies went from 10% in 2010 down to 2% in 2019, as shown on the left side chart. The chart on the right side showed during the same time period, the average forecast peak sales per asset shrunk 50% from $816 million US in 2010 to $376 million US in 2019. So on the one hand, the The cost of bringing a molecule to market has increased significantly, while on the other hand, forecasted peak sales have dropped almost that same percentage, making the entire financial equation that much more challenging over time. As if the market ecosystem challenges weren't enough, the actual development phases have their own associated challenges. At the highest level, the path to commercialization of a new monoclonal antibody has four main steps. They are early discovery and research, followed by development, next, the clinical trials phases, which seek to establish safety and efficacy, and finally, assuming everything is successful, scaling to the appropriate volume for the intended commercial target market. Let's take a deeper look at the associated challenges by development phase and opportunities and considerations at a high level. The objective of the early discovery and research phase is to identify a candidate mAb molecule to be able to enter first-in-human trials. The goal here is to optimize the R&D efforts at the bench while stretching investment dollars made, while also trying to drive the timeline down. This is where the hard science is being done. However, as my process development colleagues tell me, not all monoclonal antibodies behave as well in real life as they do on paper, and the implications of each molecule's inherent scalability, predictability, and stability will carry over into the subsequent process development phase. Some opportunities to overcome the challenges in this phase include leveraging new and emerging technical innovations to improve precision, increase throughput, expand parallel processivity, and automation. For those performing technical transfers to a CDMO during this early phase, performing comprehensive gap assessments can inform on both raw material and capability concerns, which may bring about further technical evaluations. Digitizing data transfer at this early stage can improve accuracy while reducing human error and effort. Lastly, leveraging platform cell lines and/or platform processes can help get new molecules to and through the proof of concept phase. For many, leveraging a CDMO and the experience they gained over multiple iterations is an invaluable asset. In the development phase, you've proven the molecule is safe and the candidate is able to move forward. Now it's all about mastering the recipe, pulling the various levers to build a robust, informed, productive, and scalable process for healthy cells and maximal monoclonal antibody yield in upstream and downstream processes leading into phase 2 and phase 3 trials. The main challenge is to conduct the full design of experiment efforts to generate sufficient data at the process-specific and product-specific levels. Another relevant consideration at this stage may be determining facility fit, meaning capacity, quality systems, total capabilities, and availability for clinical trials, material, and beyond. This to avoid unnecessary switching in sites or suppliers stemming from capacity limitations. Some opportunities to overcome the challenges in this phase include being able to rely or focus on critical platform knowledge, or those elements which are largely conserved or very well known. In other words, knowing which variables you can and should adjust. There's also opportunity to leverage a number of technically advanced high throughput automation systems, which can drastically accelerate key steps, which we'll mention a little later. Lastly, it's important to identify and characterize key ranges for manufacturing to avoid deviations that would cause failures down the line. In the trials phase, the goal is to achieve the trial objectives for effectiveness of the molecule in question. However, it's critical to consider the impact of scaling production to the yield and quality requirements of not only the number of patients being dosed, but also for resupply needs in advance. The question of how much material is needed is usually front and center, but having a manufacturing partner who can effectively match potentially changing clinical demands is a great safeguard. Some opportunities to overcome the challenges in the clinical trials phase include having a responsive manufacturing partner who can coordinate raw materials via exceptional communication and forecasting. It also helps having a partner whose buying power and scale can be leveraged to ensure continuity of critical supply. The number of bioreactors, total capacity, distribution of bioreactors in various geographies helps provide the ability to adjust production based on clinical demand and/or changes in demand due to dosing requirements. Lastly, having a reliable and integrated partner for clinical trials and logistics support, which encompasses patient recruitment, patient management, distribution, and clinical packaging, is a smart approach. Being able to launch a commercial product to meet patient needs is the true end. However, achieving and maintaining commercial quality and scale presents its own challenges. The process must be stress tested, and critical parameters must be fully characterized, validated, and controlled at production scale and at sufficient number of batches to support BLA filing. Post BLA, continuous process verification must occur to ensure ongoing quality, ensure compliance, and enable continuous improvement. Opportunities to overcome challenges in this phase include standardized templates and approaches, standardized training regimens, site-wide alignment across sites for documentation, controls, and SOPs to reduce timelines while improving overall cohesiveness, and leveraging surrogates instead of actual product or process solutions. Before we get into the phase-specific applied principles and case studies, it behooves us to address some key regulatory considerations as precursors to long-term success, particularly in the area of first-in-human trials. In this section, we will address what should be the right approach toward regulatory concern. What are some benefits from having an early and strategic regulatory plan? And how does advanced communication with regulatory agencies help with critical first-in-human trials? For small and emerging pharma companies in particular, the CMC or chemistry, manufacturing, and controls activities are sometimes siloed. Planning a well-vetted CMC strategy, particularly for first-in-human dossier preparation and entering the clinic, means CMC regulatory must be an early and engaged partner in all aspects of drug development discussions, from preclinical to clinical. Proactive CMC planning helps to progress through the subsequent phases and towards licensing and marketing. It's important to keep the patient and clinical experience in mind. For example, consider what product changes could be clinically meaningful when transitioning from the toxicology batch to the first in human batch. Those manufacturing changes that alter impurity profiles or affect potency, and these outcomes begin to build product knowledge to inform future product development stages. The pressure to be nimble requires having a farther horizon for manufacturing development inclusive of a regulatory strategy, which can help with identifying early versus later data requirements for the dossier, mitigating bottlenecks as the clinical program expands to later phases, as well as expanding to new jurisdictions and understanding regulatory nuance and difference, and also shaping conversations with regulators. For example, having transparent, productive discussions about proposed comparability approaches. Regulators have mechanisms to meet with sponsors in advance of a first in human trial submission. So why should you do it? One, regulators can get to know you, your company, and your molecule. Two, it's an opportunity to gain feedback, formal feedback to CMC strategies and future development plans, while benefiting from the regulator's knowledge, expertise regarding regulations, and experience based on other similar products, which may be able to help identify and avoid unnecessary development studies. Three, it can allow for alignment on content and expectations to avoid CMC clinical holds and costly delays to reach certain milestones. This is particularly important for novel products and new manufacturing processes to get early buy-in and approval. Some best practices to take are first, regulators know the science, but they don't know your product or your process. So the CMC dossier must answer the why early specifications were set, why purification steps were chosen based on expected impurities. Preventing gaps in the story helps ensure a successful dossier. Second, write the CMC dossier to fit the broadest possible audience, keeping the dossier jurisdiction neutral, allowing it to be used for other jurisdictions to facilitate future lifecycle flexibility. And thirdly, plan to include key CMC highlights and plans in the pre-filing meeting to advise what's coming next in the dossier. The best time to seek regulatory feedback should take into consideration your overall timeline to enter the clinic and engage regulators early enough to allow time for consideration of their feedback and make adjustments in your development plans. Now let's jump into the early phase development, going from DNA sequence to IND or IMPD filing. In this section, we'll look at what principles to employ to accelerate early development and how Thermo Fisher Scientific's Quick to Clinic service offers that to our clients, how technology can help reduce risk, and how risks taken need to be relative to the overall reward. Before we get into the mechanisms to accelerate early development timelines, we ought to understand the headwinds facing a candidate monoclonal antibody molecule and understand why a healthy pipeline and urgent timeline are both critical. Based on a 2021 report co-authored by Pharma, and QLS called the 2021 Clinical Development Success Rates 2011 to 2020, mAbs had an overall likelihood of approval of 12%, meaning from phase 1 to final FDA approval rating, approval during that 10-year period, as shown on the left chart. Other modality likelihoods of approval range from as low as 5% to as high as 17%. Looking phase by phase, shown on the table on the right, mAbs had a 55% probability of success from phase 1 to 2, a 34% success rate from 2 to 3, and a 68% rate of success from phase 3 to NDA BLA, with almost 100% success rate for final approval from NDA BLA. The average time to receive an approval during that 10-year period was approximately 10.5 years, with faster candidates taking approximately 9 years and slower ones averaging about 12 years. High fallout rates and lengthy development timelines necessitate a large and continually fresh pool of candidate mAbs if the market is going to see material additions of new therapeutics over time. With 90% of all drugs failing to make it through clinical trials, along with decade-long development and approval cycles, innovators are trying diligently to accelerate the first step of getting to the clinic with a new mAb candidate. But how can that best be done? We believe three main principles provide a proper framework. At a high level, those are: one, leverage advancements in technology to accelerate development, specifically increase productivity, automation, precision, control, and quality. Two, careful construction and parallelization of drug substance and drug product related activities and timelines, as well as pre-qualification of common vial formats. And third, leverage prior process experience, proven platforms, established protocols, and templated processes to reduce time while ensuring risks taken are relative to actual reward. Thermo Fisher's enhanced Quick to Clinic for biologic service is based on those principles and gets clients from DNA to IND or IMPD filing in less than 13 months for IgG1 and IgG4 monoclonal antibodies. We believe 13 months is a healthy balance between speed and appropriate risks. Clients provide us the starting DNA sequence, and in turn, they receive released drug substance and drug product, which includes 1 month stability data, quality reviewed reports, and a CMC regulatory dossier ready for filing. Along the path, we leverage the Gibco Freedom XP-CHO-S commercial ready cell line to provide a 4 to 5 month cell line development timeline to final clone selection. Also, a dual vector system to control heavy and light chain expression ratios, cGMP host cells banked in chemically defined media, free of any animal-derived components, and a favorable licensing program with no royalty payments, exit fees, clinical milestone payments, or regulatory support fees, which helps to accelerate the project initiation and set up our clients for long-term financial success. We perform formulation screening rather than full formulation development to accelerate the process. Additionally, utilizing our own mAb workflow or multi-attribute method, which is an LC-MS-based protein quality attribute quantitation platform, allows for more information to make better and faster decisions in the upstream process development steps, but more importantly, creates a solid analytical foundation for critical scale-up activities on the path to eventual commercialization. Another key benefit is a seamless drug substance to drug product handoff process, leveraging Thermo Fisher's experience and expertise in sterile fill finish at established manufacturing sites in North America, Europe, and soon to be expanding into Asia. Optional clinical trial packaging and labeling can be supported as needed. From transfection to filing, the green highlighted steps shown here are the optimized, constructed, and adjusted steps to speed up the timeline to the clinic while balancing risk and quality. We leverage a host of enabling technologies in cell line and cell culture development and monoclonality assessment, which we'll cover in the following slide. Analytical method establishment is pulled forward as early as possible, and use of transient material gives speed whenever appropriate. We use a strategic mix of binding ELISA and the mAb workflow and other platform methods for analytical method establishment. Co-qualification during analytical development between drug substance and drug product sites enables concentration and ID assays to be done together rather than separately. Parallel execution of upstream process development and cell line development workflows collapses the overall timeline. We have confirmation runs and toxicology batches performed at the same time to understand robustness of the process while generating tox material. And lastly, coordinated shipment between drug substance sites and drug product sites help to reduce the inherent lag time. Advances in bioprocess technology include higher expressing cell lines, improved bioreactor performance, increasing single-use capabilities at ever-increasing scales, and higher throughput screening tools. These have all helped maximize output while reducing effort and time on the front end. We leverage the Berkeley Lights Beacon optofluidic platform, which allows for single-cell clone in situ antibody titer assessments, as well as the Sartorius AMBER 15 mini bioreactor, which allows for parallel growth and productivity evaluations. As previously mentioned, we employ our own LCMS-based analysis and the AMBER 15 bioreactors in parallel with the CLD steps as we scale up to cGMP batches to develop the best growth conditions while ensuring stability and product quality. As such, we are able to shave 6 to 8 weeks of time versus traditional methods while still having confidence in the lead clone with a high level of simultaneous process knowledge. These attributes contribute to a client being able to accelerate a molecule from concept to clinic. However, to gain some of that speed, some flexibility is necessarily sacrificed. As such, this offering only focuses on early phase first in human, supports only liquid-filled vial formats, and follows a proven but limited flexibility template of both process and documentation. I'd like to share a quick case study of one of our quick-to-clinic clients. In this case, the client's a late-stage clinical biopharma with operational footprint in the Middle East. The area of focus centers on the discovery, development, and commercialization of first-in-class cancer treatments. Their candidate molecule was a traditional IgG1 mAb, which made it a perfect fit for our offering. However, this client was pretty conservative and also relatively new to biologics at the time. Our team responded quickly, presented them the benefits of leveraging our solution to get them to clinic fast. We held face-to-face meetings to review our capabilities and present the benefits of our platform solutions, which piqued their interest. They signed on to our initial quick-to-clinic offering, we'll call it version 1.0, and also added elements of our quick-to-care or fully integrated offering, which I'll speak to next. Thermo Fisher Pharma Services Group provided them solid insight on clinical strategy and timing of studies to support the pre-IND meeting. This was back in 2020. This client fully leveraged the global logistic footprint of our drug substance and drug product network and is now currently on our version 2.0 quick-to-clinic offering, and presently the IND is pending second half of 2022 completion. Now, despite all the emphasis on speed to clinic, too much of a good thing can be a not so good thing. As such, we like to present 3 pitfalls to avoid on the road to IND and IMPD. Speed shouldn't be prioritized over other critical success factors. Time pressures can result in missed opportunities, poorly considered platform selection, inadequate material for scale up or clinical trials, or even unsatisfactory analytics data for IND and IMPD applications. Each of these risks have become significant financial, time, regulatory, or logistics problems at the point of application or beyond. To avoid jeopardizing your IND IMPD while still maintaining aggressive timelines, consider the following strategies. One, leverage integrated drug substance drug product programs to shave time without sacrificing other key metrics. This means taking advantage of platform products and standardized offerings where relevant and available, achieving efficiency through high throughput automation technologies, and most importantly, parallel and concurrent characterizations and analytical method qualifications, especially with a partner who has had demonstrated success and experience. Two, thoughtfully consider risks and potential future impact on filing, on scale up, and other future goals. Each accelerated step skipped or altered to save time should be fully evaluated. Smart and thorough toxicology testing, confirmatory satellite runs, standardized protocols, and advanced screening technologies can enable smart yet rapid progress with relatively low risk. Lastly, work with experienced partners who understand process development and scale-up challenges. Experience gives them invaluable insight and credibility, especially if they participated in the full development process many times for a variety of molecule types. Any partner should be one with deep experience in both traditional and accelerated process. This to protect both parties from unrealistic expectations and unforeseen quality concerns or delays. Filing the IND or IMPD quickly shouldn't mean putting the blinders on to future scale-up, scope, analytics, practical costs, or logistics. These may seem like far-off factors irrelevant to early decision-making, but they can make or break the success of a drug product during clinical testing or commercialization. To reduce risk of future platform changes or forced cell line reevaluations, scalability should be considered sooner rather than later. Ways to increase scalability success include harnessing robust platform processes. Starting with a strong platform process selection is an essential step. Working with a development and manufacturing partner with a robust platform process that identifies components that can be standardized can remove the burden of starting from scratch for each drug substance. Ideally, a standardized platform process should work reliably and predictably from inoculation to fill finish, relying on vetted technologies with performance history. Our own standardized platform features harvesting, viral inactivation, chromatography, filtration, buffer, fill finish, and storage practices and equipment that meet and or exceed industry standards. Starting with a thoughtful framework leads to fewer surprises after approval in terms of materials, scalability, and quality. Also, consider the fees, payments, and licensing implications. Understanding these costs upfront and how sustainable they will be at the point of scale-up is essential to an economically viable product. Consider how molecules are licensed. Are there royalties or clinical milestone fees? Is regulatory support included in the license? Do the host cell research rights allow for contract development or manufacturing? Are there exit fees? Are there opportunities to reduce fees through upfront payments or other options? Pitfall 3 occurs when the focus is only on phase 1 trial needs. However, neglecting to follow proper testing and documentation can be an obstacle to swift IND or IMPD approval or limiting the potential product knowledge learned from early research studies. Early phase clinical work has very different requirements than late phase and commercial use in terms of process efficiency, production costs and timeline, supply, yield, scalability, product delivery, performance, and storage. What's sufficient for small-scale pharmacokinetic and pharmacodynamic studies and first-in-human proof of concept studies may not be practical to scale or maintain in future clinical development stages or after marketing authorization. Identifying critical requirements for filing and phase 1 initiation and how production needs will change as the molecule advances allows for early planning to provide an advantage for approval, transfer, future research, and commercialization. Companies can proactively support IND-IMPD approval and downstream transitions by having experienced regulatory experts document and gap assess all CMC elements for IND submission. As we stated during the earlier key regulatory considerations section, it's important to work with knowledgeable and experienced CMC partners and to employ those best practices mentioned. Working with a partner with capabilities beyond drug substance and drug product development and manufacturing. Think ahead to a partner who can readily transition you out of phase one into the next phases of clinical development and larger trials. A contract partner with experience in all stages of post-manufacturing services can make the long-term experience more streamlined, more predictable, and ultimately simplified. With an eye on future commercialization, it's important to think about label design, final packaging, storage, and distribution, and logistics. So what about the rest of the development phases and what principles are important when moving to late phase and commercial launch? What are some of the challenges facing innovators as they navigate this phase? Are there ways to reduce obstacles and hindrances on the way to market? Historically, innovators hop from vendor to vendor, from phase to phase, to essentially shop for the best phase-specific partner. That approach meant multiple service and quality agreements to review and negotiate, as well as multiple invoices to reconcile. It also meant no single entity was accountable for the overall program. Each vendor was responsible only for their part of the equation. Technical transfers and development phase transitions would necessarily have to occur between different companies and sites with very different levels of communication and collaboration. The flow of regulatory information from phase to phase and company to company can be burdensome if not centrally managed too. Transportation of materials would have to be organized between different CDMOs, manufacturing sites, clinical trial sites, and distribution network locations. Innovators would expend considerable effort, time, and resource to coordinate all the disparate steps involved. And in the end, the innovators carried nearly all the risk. Whereas the enhanced quick-to-clinic for biologics offering mentioned previously is geared for speed and early phase only, our quick-to-care offering covers the entire continuum from early development through late phase clinical supply into commercialization. It's our fully flexible and fully integrated offering, which changes the historically fragmented approach where individual vendors or CDMOs are only responsible for one part, to one where an innovator can effectively start here and stay here from concept to patient and gain a myriad of benefits from having a single solution provider. The program's success is due to a comprehensive and integrated drug substance to drug product plan, supported by a single program manager dedicated to the project who acts as the client's point of contact to ensure full visibility and control. The program manager integrates all of our capabilities, including the power of a large global network of subject matter experts, facilities, and capabilities, and ensures communication and governance to ensure rapid decision-making, raising awareness of issues and proper oversight. Operational efficiencies are gained in terms of technical transfers, being able to leverage shared documentation, as well as internal process owners, common or shared equipment knowledge, approved vendors are universal across all our sites, and greater visibility to allow for dynamic changes in capacity or localized supply challenges. In short, coordination, commitment, and communication are working to ensure accountability for the client, regardless of if they're a large pharma customer or a two-person virtual biotech. The program also offers other benefits, providing a single service agreement, harmonized quality agreements, integrated and combined proposals for efficiency, and still saves an average of 14 weeks of time from start to finish. The Quick to Care program covers our large molecule and biologics portfolio, but also extends to small molecule drug substance, oral solid dose, sterile drug product, and most recently, our cell and gene therapy services offering too. One unique benefit of Quick to Care is our clinical supply optimization service, which works with clients to define the clinical supply strategy, which includes forecasting, dynamic reforecasting, experienced supply chain managers in different geographies and therapeutic areas to ensure clients have the right medication in the right place with the right label. Total transportation management is another benefit, which provides a global network of carriers, dispatch control services, a global network of cold chain storage facilities, customs and regulatory support, insurance, and full inventory control to ensure end-to-end supply. If you think quick to care is only for larger, more established pharma customers, think again. Quick to care is ideal for clients who have minimal internal support, who lack expertise, who have complex trial designs, who need flexibility, and for those who expect variable resource demands throughout the trial. Here's a quick case study on one of our quick to care clients. We had two clients who were working together, one headquartered in Europe, the other in Asia, both focusing on cancer biologics. The target molecule was a traditional monoclonal antibody, and one of the partners had been working with Thermo Fisher Scientific and had completed phase 2 development. They actually tried shopping for another CDMO for phase 3, but ultimately came back to Thermo Fisher. The two companies had divergent company cultures, needs, and expectations. CMC strategy was a critical need for both, and a partner with a global approach was extremely important to them. They leveraged the quick to care offering as an ideal solution, including 3 sites in our network: Princeton, New Jersey for development, Groningen in the Netherlands for drug substance production, and Ferentino, Italy for drug product production. The governance and collaboration benefits of our program streamlined total communication between clients and sites and allowed for clear escalation when necessary. Early on face-to-face regulatory alignment facilitated a streamlined approach, and despite the early days of the pandemic affecting travel, PPQ was still accomplished and successfully navigated. The BLA was submitted in the second half of 2021 and is now pending health agency approval. Before I summarize the main points covered today, I would like to express a debt of gratitude to my colleagues in the Pharma Services Group, as well as those in the Bioproduction Group, who have provided me this opportunity to speak. A very special thanks to my colleagues in the Bioprocess Sciences Group, the Scientific and Technical Affairs team, the Process Development team, as well as my partners in Global Marketing for helping with all the slide content. And finally, thank all of you for your attendance and attention. In summary, monoclonal antibodies have filled and will continue to fill a very critical role in the global therapeutic pipeline. The pressures driving speed to clinic and speed to patients are diverse and challenging and require careful balance between risk and reward. 3 elements to consider are: 1, how and where you can optimize in the early stages to get to first-in-human trials, by leveraging technology to accelerate development while reducing risk and considering careful construction and parallelization of drug substance and drug product activities. 2, remember the 3 pitfalls: IND, IMPD at all costs, IND, IMPD over everything else, and focusing only on phase 1 supply needs. And 3, for late phase, bear in mind, what is your clinical supply strategy? Do you have the transportation management piece locked down? Do you prefer managing a decentralized network of vendors and suppliers? Or should you go with an integrated solution provider? Finally, speed to successful IND, IMPD, and beyond is achievable when companies leverage integrated programs, adopt robust platforms, and align with partners offering full service support and experience in regulatory submissions. Thank you.